चेक करो लाइव हो गया क्या? Now we are live.
Shall I start?
Start करें Ravi?
Yeah.
Yeah. Yes, we can start now. Yeah.
Good morning, esteemed fellows and the audience. It's a matter of pleasure for us that today we are here for the first seminar in the series of the 75th year
Ravi,
of independence.
live हो रहा है क्या?
Amrutotsav. We have with us the president of INSA, Professor Chandrima Shaha, and the first speaker of this series, Dr. Tapas Kumar Kundu, who is director of the CSIR CDRI at Lucknow, Central Drug Research Institute. He's a known authority in the field, and it's indeed a pleasure to have him as a first speaker in this web seminar series. A decision was taken that all across the country, the scientific institutions would celebrate the 75th year, that is 2022, as Amrutotsav or Amrit Mahotsav. And this is to remember that we became independent in the year 1947, on 15th of August, as we all know. Science has been not a miracle, but this has been a solution to many problems of the country. And this is an opportunity for all of us to help us realize how important science is in today's world. And the COVID-19 pandemic has shown it again and again that without science, it's very difficult to survive and sustain. Today's lecture is the first in the series, as I said, and as has been said on many occasions, science has provided humanity solutions to many of the problems of humanity, be it hunger, be it diseases, be it illiteracy, superstitions, or many such social evils. And I would request Professor Chandrima Shaha, a renowned scientist, a renowned life scientist, former director of NII, and the president of Indian National Science Academy. Now, please welcome and introduce the speaker of the day.
Thank you, Dr. Nautya. I once again welcome all to our first webinar in the celebration of Amrutotsava. First, let me say that how pleased we are to have Professor Tapas Kundu with us, a very accomplished scientist. And it is really a pleasure to introduce him. I mean, his CV is quite large, but I'm going to shorten it for the interest of time. But one can check his credentials on the net. Now, Professor Kundu did his PhD from the Indian Institute of Science, Bangalore. And following his PhD, he had a short stint as a visiting foreign research associate at the National Institute of Genetics in Japan, Mishima, Japan. And this was followed by a postdoctoral fellowship at the Rockefeller University in New York City. And then he joined JNCSR Bangalore. Currently, he's heading the Central Drug Research Institute of CSIR as director since August 2018. Professor Kundu has created an excellent blend of epigenetics and chemical biology in context of cancer diagnostics and therapeutics. He has not only discovered several therapeutically relevant inhibitors of acetyltransferases, but also has shown that these inhibitors are highly effective to reduce cancer growth and efficiently in various model systems. I'm sure we're going to hear some of that. He's the recipient of several awards. I'm going to mention only a few, noteworthy among which are the Shanti Swarup Bhatnagar Prize from CSIR, the India Reliance Industries Platinum Jubilee Award, Sir J.C. Bose National Fellowship, G.D. Birla Award for Scientific Research, Ranbaxy Research Award in the field of medical sciences, India Innovation Award given by the Merck Manipur, and Silver Jubilee Professorship of JNCSR and the Om Prakash Bhasin Award, and also many others. He was instrumental to establish the Asian Forum for Chromatin and Chromosome Biology and Chemical Biology Society in India. Besides fundamental research, Professor Kundu is also involved in teaching and organizing science outreach programs. His popular lectures on genes, disease, and therapeutics have benefited many high school and college students. I'm sure today's lecture is going to be similarly exciting and interesting for the audience. And I welcome Professor Kundu to deliver his lecture. Thank you very much.
Thank you, Professor Shah, for very kind introduction. And I am really grateful to INSA for giving me this unique opportunity to inaugurate the AMRUTOTSAVA for our 75th anniversary of our independence. And I also thank Dr. Nautiyal and Dr. Shima Mandal for organizing this talk. Today, I'm going to talk to you about a subject which is all around us in every moment of the life, every steps of the life. It is actually, affecting our life and our health, and that's the reason it has become the important target for therapeutics as well as the management of life, I would say. So the subject which I'm going to talk about is the epigenetics. Now, a very simple concept. What I would like to talk to you is the epigenetics concept, that we all start our life from a single cell. And if you see the life from this single cell, I just try to get the pointer. Laser pointer. Okay. If you see we all start our life from the single cell, and this cell, the pluripotent cell, the zygote, when it differentiates, it makes the different kinds of cell: blood cell, kidney cell, brain cells, and muscle cells. But you will see each type of cells are different by looking, by function, by gene expression. Everything is different. Now, a very simple question, if I ask that it originated from the same cell, they have identical DNA sequences, then why it is different? Why it is functioning differently? Why it looks very different? So the gene sequence is not enough. Gene sequence is not enough to determine. And if you see the Waddington's, this downhill process, and it is coming down, and it is making each type of the cell. So the question, and it is now, earlier we knew that we cannot go back, but now due to the discovery of Imanaka factor, we know that we can also go back to these steps. Anyway, so the very important thing is that the gene sequence is not the determinant of the gene expression. So something above the gene, and that is epigene, and that is epigenetics. And so this is the fundamental concept. I gave you a very nice, so sometimes back, an article published in Times magazine, which had shown that your DNA is not your destiny. So if this is your DNA dictionary, this dictionary is not functional until your DNA is properly labeled and properly made functional. A very easy example, if you see these two mice, one is yellow, the other one is gray, and it's a thin and brown, sorry. So if you see their gene sequence, it could be identical. But if you see their look is very, very different, maybe their behavior also very different. And how is it possible? It is possible because the genes are modified. In this case, it is differentially modified, it is methylated. And that's the epigene. That's the epigenetics. So now, if I go a little deeper, all our DNA in eukaryotic system, in mammalian system, is actually packed in a nucleoprotein structure we call nucleosome, and the threads we call chromatin. And each chromosome in our cell are actually present in a resting cell as a independent chromatin territories. They are folding and unfolding, which is responsible, which is the epigenetic phenomenon is responsible for that, is actually controls the gene expression. The gene expression depending on what? Depending on the signal. What signal? Everything is signal. We are happy or unhappy. What food we eat. What bacteria we are infected. What bacteria we are carrying with us. Which virus is attacking us. Everything affects the signal of these genome folding, unfolding. So the song of life, which you see is written in a code in our DNA, is only will be functional when this DNA is properly decorated. Not only in the DNA, in the DNA or also the DNA associated protein. So these modifications together is actually sets the epigenetic concert. So it could be the DNA modification like methylation, hydroxymethylation of the DNA. It could be DNA associated protein, that is the histone modification, or the genome folding can also be altered by the long non-coding RNA or any other small non-coding RNA. So these are the epigenetic tools. And how is it function? How is it connected to light? So if you look at Mendel's phenomenon, Mendel could see the nature, but Mendel could not describe at that time the nurture. And this nurture is called the epigenetics. I give you a very easy example here. You can see in the screen, this is the mother, okay? And this mother, suppose she is smoking and she has a female baby inside her. Her smoking is going to affect the ovary of this small baby. And this effect, what she is creating is not the genetic effect, epigenetic effect. And that can go even up to third generation, fourth generation, fifth generation. So that means without changing the genetic set, just by gene modification, even can go from one generation to another generation. Of course, epigenetics are most of the time is within you, but whenever it is affecting the germ cells, it can also become transgenerational. So this is one of the behavior. So who is the most important person in your life? Most important person in your life is your mother. Because I show you a picture here by the famous Indian painter, Jamini Roy, and you see this affection of mother and this confidence the child have on the mother is one of the important epigenetic, the tools or one of the important epigenetic means. Because the mother, she has not only shared half of her chromosome and cytoplasm in your first one cell stage, you have also grown up within her. Her everything, her food habit, how society has treated her, how she was psychologically happy or unhappy. Not only that, how she took care of you in your first few years has everything affected your epigenetics and has everything, what you are today is largely because of your mother. And this is not what I'm talking a very emotional fantasy. No, I'm giving an experimental evidence. If you see these small pups, so these mice or rat, if these pups get very good care from the mother, then these pups, the genes in their hippocampus, genes which is required to face the struggle, to become the confidence. So these genes will not be methylated, okay? But if these babies, these pups don't get a proper care from the mother, you will see they are, because of this trace, the hippocampal genes will be methylated. And these pups, when they will grow up, they will not be able to face lot of the struggle of life. And they will be inconfident, they will be aggressive. And if you swap the mother, the same pups which were become like these, they are becoming, they become aggressive and all, they become inconfident, they will be much better. So this has been shown experimentally. Yes, it is true. So what does it mean? It means to you that the genes, yes, it is important, but the epigenes is even more important, and it is directly connected what you have received and, besides the gene from your parents. So, but the epigenetics can also change because of the aging process and how you have aged, that also. If you look at the small baby, when she will give everything to this society and she will become Amma, she will be maybe in the one corner. And the in between, besides the chromosome breaking and joining and all, there will be a lot of epigenetic changes in her. And these epigenetic changes could be, very simple, the acetylation of the histones present or methylation histone presence associated with the gene, besides the DNA methylation. And what all other way epigenetics can be affected. Today, the terminology is coming with social epigenetics. Social epigenetics means human being is a social animal. And this human being has a very close interactions with the nature. Human being has a different kinds of, food habit, different kinds of habit. And human beings are subjected to pollution. Not only that, human being also subjected to the several, the trauma in the life. And everything affects your epigenetics. And that gets reflected in your downstream life. So the social epigenetics has become a very important thing. You see, I gave you a picture. This is one of the picture of a street drama in Mumbai city. So this is the street drama a voluntary organization organizes to show the, the violence to women. And this is happening every nook and corner of the world, that this kind of incidents. These incidents not only affect the psychological, the downstream life of women, it can also terribly affect the epigenetic cassette and create the depression and create so many other problems in the life. So not only that, so the stress and trauma, it is proven now, causes so many other problems like aggressive behavior, the hostility, the depression, the so many other proactive aggressions, and these all are connected to the epigenetic changes and epigenetic modifications. And of course, this is directly connected to the hormonal. Actually, the upstream is the hormonal function, followed by the downstream. But the question is, since the epigenetics is reversible, and that's the reason there is some hope. I show you a nice small video here. So this, you will see that this one second. So I just video. So this is a story of you.
Started with DNA. Your DNA was a genetic blueprint that most people turns out it's unfoldable.
So if you have a good life choice, then you see this is what it happens. You get a more energy, a better sleep, your BDNF in the brain is expressed better, and thereby you become victorious. But good habit cannot sustain for a long time. If you have a bad choice, then it happens. You have a big burger, less sleep, a lot of other things, and you see the pouch has come out. Okay. So these are the genetic characters. And these are the epigenetic characters. So this is very simple video to you. And this it shows that even though so many things, since you are a single cell, it affects your epigenetics, but it can actually, it can actually be reversed with your habits, or it can actually be controlled with your habits. And this could be the exercise, this could be very good nutritional food, this could be your environment, this could be your emotional health, that is the meditation and all, and of course the different kinds of antioxidant, anti-inflammatory, the foods and all things which you take. Now, all these things I'm saying you that the bottom line, what I'm trying to tell you, that since you are a single cell in your mother, since you are in your early childhood, as you grew up, as your different behavior, your environments, all these things are changing your epigenetics and thereby gene expression. So all these are actually acting as a signal for you. And does it have any scientific basis? Now we know very well that the diet, the environment, the microbiome, all actually affects your metabolism. And the metabolites, we will find out very important metabolites, which comes from either from the diet, nutrients, non-nutrients, or it comes from the metabolic pathways. You will see like NAD, FAD, alpha-ketoglutarate, S-adenosine, methionine. These all are fundamental source of epigenetic modifications. Their concentration, their presence in the cell, their outburst in the cell, all directly affect the gene modification, gene function, and thereby your life in a larger context. So if I take you a very simple example, that is not the DNA modification, the DNA associated protein like histone modification. If I take the lysine acetylation, I'm talking just one modification to you, and you will find out that the lysine acetylation is very, very important epigenetic modification for your life. And this can be acetylated by lysine acetyl transferases. It can be deacetylated by the lysine deacetylases. And this acetylome balance has a very important role to play in your life. These are different acetyl transferases human beings possess. If you take one of the important category is P300, CBP, is a very large protein, and I'll come in a moment. So this is the acetylation homeostasis. When it is deacetylated, genome is compact and repressed. When it is acetylated, the genome is open and it is functional. It can synthesize. Now this opening and closing is very, very important for your very timely gene expression. And if this timely gene expression is not happening, that can cause so many diseases, cancer, inflammatory disorder, etc. And in the other hand, it could also be connected to the neurodegenerative disorders. So I will talk to you down the line is the little bit about the cancer connections and the neurodegeneration connections, and whether we can target these alteration of this modification for the therapeutic purposes. And it is evident in the cancer that there are several important histone modifications. These are the nucleosome, this is DNA. These are the different histone octamers present in our genes, and the histone tails are outside. They are subjected to large number of modifications, acetylation, methylation, phosphorylation, and which directly affects the gene expression and thereby the disease context. And if you see that depending upon the cancer we are talking of, depending upon the disease state we are talking of, the different lysine residues can be upregulated or downregulated, and that may be caused by the different enzymes. So, and this may be connected either. So the histone modification, if you see, it could be hyperacetylated, it could be hypoacetylated, depending upon the cancer we are talking about. Okay. And so this could be like, for example, in the breast cancer, there is a type of breast cancer, you find out the hyperacetylation. Oral cancer, largely it is the hyperacetylation. There are some cases it could be hypoacetylation. So either the acetyltransferase, deacetylase, could be the target. I give you just a patient sample data here, just to show that in the oral cancer, we find the huge context of hyperacetylation in the tumor. The more gray is the more hyperacetylations. So you see here, just I will show you, I take the pointer option. So if you see here, the acetylation is very high in the patient sample. And not only that, the acetyltransferase, which gets self-acetylated, that is also very high. Self-acetylated one is more active. And the protein which affects the acetylation, like GAPDH, nucleophosphine 1, they are also overexpressed because they induce the P300 function. Overall, in the oral cancer, caused by largely by the tobacco, has a connection to the hyperacetylation phenomenon. So when this hyperacetylation is there, the simple question, if we along with the other therapeutics or alone, if we can inhibit the acetyltransferase activity, will we be able to control the cancerous growth? That's the question. Over the period, both my laboratory and all over the world, people have discovered several inhibitors of acetyltransferase of different classes: natural, synthetic, semi-synthetic, and it has been tried for the anti-cancerous growth. And here I show you one example, the molecule called CTK7A. It's a water-soluble, broad-spectrum acetyltransferase inhibitor. And as you see here, it is actually acting as it is reducing the cell growth, cell mobility, and it's actually reducing the tumor growth also very effectively. And if we see that it is even in the tumor, you see blue color here, the treated one has become the less acetylations, the one which is growth has reduced. So this is the hypothesis. It shows the phenomenon that yes, tumor growth can be controlled by the acetyltransferase inhibitor. We tried, this is just to show you this molecule become patented. It is now as an academic reagent, Smart Millipore is selling it. It is available, but it has given us a concept that yes, if you use these, the hyperacetylation of the enzyme comes down, the growth rate comes down, and this could be a very important, if not alone, but in combination, a therapeutic molecule. It has been shown even in the animal model system, in the bioavailability, in some type of prostate cancer, that yes, this can be used as an inhibitor to reduce. Later on, a few natural compounds like deuteoline, which is a P300 specific inhibitor we have tried. We have shown that the acetylation is inhibited, and we are also shown that this is also working as a cell proliferation inhibitor, and we showed that the tumor growth in lead comes down. So this gave us a very important clue that the acetyltransferase can be targeted. Now there is a company now which has established in the United States called Lysine, and they are solely looking and a couple of compounds are in the clinical trial. Our is not yet. Is solely looking at how acetyltransferase can be targeted for the anti-cancer therapy, anti-inflammatory therapy, and also antiviral therapy, which I will show you in a moment. But the epigenetic modification, it does not work alone. It works in conjunction with other modifications, because the epigenetic enzymes, for example, the P300, along with the arginine methyltransferase PRMT4 or COM1, they work together. So there is an interlink between the epigenetic modification, and that is what the epigenetic language is established for gene expression, upregulation or downregulation. And this language in combination with the methylation of the histone and the DNA, acetylation and phosphorylation, it work together. So the scenario is much more complex than what we think about. It's much more complex than gene function. However, we wanted to see that in these oral cancer sample, when hyperacetylation is happening, whether any of the histone methylation mark also changing. And most remarkably, what we see that histone H3R17 asymmetric dimethylation. You do not need to care about this terminology. You just see that one of the methylation mark is so high in the patient sample. And when you screen with different methylation mark, we find that yes, there is a very distinct change in the methylation. And some of the methylation is really upregulated. And we see that PRMT4 we checked in the patient sample, and we find that indeed the PRMT4 is very upregulated. And I'm not going to the detail. Now we have the data. We know why PRMT4 is upregulated in the oral cancer. Can we control it or not? We discovered an inhibitor which directly target the PRMT4. We are trying to explore this axis. Which axis? We are trying to explore the important axis that one hand we will be controlling the P300 acetylation. The other hand, we will be controlling the PRMT4 activity. And this dual, a molecule, maybe a single molecule, will have a dual activity, and that could really downregulate the tumor growth. Especially, it will be very useful for the non-adjuvant therapy, and especially to reduce the tumor growth for the downstream surgery and etc. So this is what we are working. And Maxwell National Oral Cancer Institute support from DBT is, has given a support to us for this oral cancer work. And with 7 different institutes, we are working together for looking this. I present you another very interesting molecule here. And I'm tempted to present this. When we are working with only one class of acetyltransferase, P300 and CBP, meanwhile, we are giving the screening of different natural compounds. And one compound we get is called embeline. And this embeline, what we find is a not P300, but PCAF inhibitor, which is another class of inhibitor, which is this PCAF is very much responsible for the muscle differentiation. We went ahead, we look into the real mechanism of action of it, why it is specific, how this embeline tail fits into the PCAF group, but not P300 group. There's a very interesting collaboration with the Indian Institute of Science and with the Singapore National University, NUS, with the Dr. Reshma Taneja's group. We show that it is inhibiting the muscle differentiation as well. We find out the different pathways, how this could be functioning through the acetyltransferase. A very interesting publication came up, but the fruitful work happened in this case in terms of therapeutic aspects when Dr. Taneja looked into the rhabdoid sarcoma. Rhabdoid sarcoma is a cancer, the muscle-related cancer, which has no therapy. And she finds that the PCAF is upregulated there. And I'll show you a mechanism how it actually creates the rhabdoid sarcoma. And in these, she finds that H3K9 acetylation, which is a very specific acetylation by PCAF, is highly upregulated. And when she uses embeline, this specific molecule, she could see that even in the tumor, this acetylation comes down. And she sees that this tumor is dramatically has come down. So that created an unique opportunity that the other class of acetyltransferase, other class of cancer can be targeted. And if you see, this is the PCAF molecule. And it is required for the skeletal muscle, differentiation because it methylates the major muscle differentiation factor MyoD and affects the muscle differentiation process, and the differentiation happens. But in the rhabdoid sarcoma, there is a fusion transcription factor, PAX3-FOXO1. This binds to the gene sequence, and this has a special affinity for highly overexpressed PCAF, and it targets the, it acetylates PAX3-FOXO1 and makes the gene's expression, which is required for the oncogenesis process. Now, when we inhibit these by our inhibitor, that can stop the oncogenesis process. That is what mechanistically we find. Now, in this case, we are trying to have, we are looking for it, and we are also very close to that. Another important inhibitor or another important enzyme targeting, that is the muscle differentiation in the methyl transferase G9. I have no time to say it here, but maybe in some next time we'll do that. So now in the protein lysine acetylation, we also thought of that could it also be used, look into the effect of it in the viral infection. And this is the work, it started with my, next door collaborator, Professor Ranga Uday Kumar from JNCASR. HIV virus, when it's a retrovirus, when it infects the cell, it goes inside the genome and it becomes the part of the genome. And thereby, the viral latency, the viral expression is directly connected to the epigenetic modification. Not only this modification of the nucleosome, but also the modification of the TAT protein. It actually connects the host transcription factors, host transcription machineries along with the nucleosome modification. And the TAT modification has become a very important target now to overcome the viral latency. Now, what we ask this question is that we try to generate an inhibitor from a natural compound. My postdoc, Dr. Montelingu, presently the faculty in the Mysore University, he generated a very interesting series of molecules from the natural compound garcinol. He made the isogarcinol, make the double substitutions, single substitutions, and one of the compound, what we call the LTK14, has become a P300 specific inhibitor, not any other acetyltransferase, and become much less toxic. Well, this was also a collaboration of Indian Institute of Science with the MBU, one of the MBU students long back. And then what Professor Ranga Uday find that when you treat these compounds to in the cellular model of HIV infection, the viral syncytia really comes down. Okay, these are the viral syncytia. You may know today the SARS-CoV virus, when they infect in a very high dose in the lung cell, basically the viral syncytia actually kills the lung cell and makes the hole. So, so even in the HIV case, this is you see here, these are the viral syncytia could be efficiently inhibited by this inhibitor of acetyltransferase. So there is exploration is possible, and this become an important, the means to control the viral expression. And that time the publication came. We have initiated again this breaking of viral latency, by using the different small molecule, which will be targeting not only the acetylation, but also methylation, not only the histone modification, but also TAT modification, which is at present going on. I go to slightly switch in the behavior part. So one of the important, the phenomenon as we grow in the life, to stay fit, lean and fit. And that has a lot of, there are some genetic connection, but it has also the habit connections. And obesity is the today's world problem. The obesity, if you see, that connected to so many things, cardiovascular disorders, stroke, diabetes, renal failure, and cancer, and so many other connections. So my lab became interested to look into the epigenetics and obesity. And we took, again, the very important route. So we find that, the adipogenesis, the process, is connected to the histone methylation, and it is driven by PRMT4 COM1. And this has a connection with the P53, the famous tumor suppressor molecule. While doing these, we try to find out the process of adipogenesis, how it can be affected by different epigenetic modification. And in this case, we targeted not the acetylation, but much smaller modification, much slower modifications is acetylation. Okay. And this is a new series of modifications is now being discovered. like, amylation, protonylation, deuteralylations, etc. And my student Adi looked for it, and he used the pre-adipocytes and adipocyte differentiation system in a cellular system. Then my postdoc Saurabh, from the garcinol derivatives, he could synthesize a molecule which could inhibit the adipogenesis process. And see that it is working in the high-fat diet mice model to reduce the obesity. Now we know the exact gene signature, we know the exact modification, even we have tested in the genetic model, and it is fantastically working, targeting one very specific acylation. And we are thinking that this could be an important molecule to take it forward for the anti-obesity, which is going on in collaboration with JNC and CDRI. And we are about to submit this patent. And the phenomenon is that when you give either the high-fat diet or in the genetic model, this acylation process induce the obesity. Now, if you can inhibit the acylation by a small molecule, we have shown the obesity has controlled quite a lot without giving any health defect to the mice. So, this shows that the epigenetic modification could be an important target to fight the obesity without harming the physical health. Okay, so sorry, this slide appears here again. I'm sorry about that. So the last part of my talk, I will take another 15 minutes. I will switch over the gear for a serendipitous discovery of the laboratory and a very upcoming thoughts about the how we tackle with the neurodegenerative disorder by targeting the epigenetic modifications. So, as you see here, that you all are listening me and you are recording. And when you are recording in your mind, your maybe long-term, short-term memories are actually forming. In this whole process, epigenetic modification plays a very important role because the memory consolidation function is directly connected, you will see, is with the calcium signaling followed by the epigenetic modifications. And these epigenetic modifications, especially the acetylation driven by CBP300 enzyme, is very, very important for the memory consolidation function. If you look at the physiological and the pathophysiological state, acetylation plays a very important role, even in the aging process. When you look at in the aging, you will find out the localization of the acetylation, the level of acetylation is tremendously altered. Look at even the depression case, the dramatic reduction of the acetylation happens in the depressed brain. And so the question comes, it can also be played with the depression phenomenon. We have done some work. Because of time reason, I will not be able to show. So finally, the entire process could be controlled by the reversible acetylation process. So how we entered in this field? Long back, my lab, at the very early stage of the lab, discovered a natural inhibitor of acetyltransferase. It's called anacardic acid, which later is basically aspirin. Later on, Eric Badet's group very recently have shown that indeed aspirin can act as an anti-cancer, and it is an acetyltransferase inhibitor inspired from this one. So this anacardic acid was a broad spectrum acetyltransferase inhibitor. And this, when we, my postdoc Karanam Balasubramanian, when he was working on this, he was not very happy with the simple molecule. He started making this molecule more complex. And he put a flutamide ring with it. And he synthesized a molecule which we call CTPB at that time. This molecule, instead of inhibiting the acetyltransferase, it activated the acetyltransferase. This was very desired molecule, but it was very, very, unexpected to us. And it actually encouraged a lot of hope in our mind that if you can activate specifically acetyltransferase, you can directly address so many important disease, especially the neuro disorders. And so these molecules become very popular in no time. We got the US patent, and unknowingly, without informing us, even though we had a patent, several companies start selling this molecule because it became a hot cake that you can address this. And we were very much interested that, as I told you, all the neurodegenerative disorders, most of the neurodegenerative disorders, is somehow connected to the reversible acetylation process, and it actually goes down. People are thinking to use the HDAC inhibitor, deacetylase inhibitor, to induce the acetylation. But here we have a tool which can directly help. Even though this molecule becomes so popular and it's become commercialized and all, we were not happy with it. The reason we are not happy with it, because this CTPB, this molecule, in spite of derivatizing and in spite of making so many derivatives, it was a very, very poor cell permeable molecule. It was not getting into the human cell that easily. And we were a bit frustrated at this time, but chance favored the prepared mind. As it happened, next door of mine, Professor Ishwar Murthy's group. Ishwar comes from our chemistry, physics, and material science unit of JMCASR. He is an expert of the nanochemistry. Ishwar was working, his group was derived carbon nanosphere. And these carbon nanosphere, basically a hydrothermal treatment of the glucose. And when his student Dinesh had given this to my student Selvi, and she finds that this is a self-fluorescent molecule. I told her, put our CTPB on top of it and see whether it can take into the cell. And we see that this nanosphere can cross the cellular membrane. By 3 hours, you see Chandrayaan has landed on the moon, and by sometimes more 24 hours, it has become a green cells. And we see that it could really carry this on and induce the acetylation heavily. The most encouraging and surprising for us was when you use these molecules subperitoneally injected the mice, we see that this could cross the blood-brain barrier. You see these yellow dots. And it could induce the acetylation in the brain. This was something really very encouraging. For a long time, we were not even believing this is possible. I talked to my collaborator in France, who is a neurobiologist. She took this molecule from us and she used it, and it was a very surprising that they also got very specific upregulation of acetylation in the brain. And then we tried to make this molecule a little bit stabilized. We took the tail out. My postdoc Pushpak Mizar from NEHU, he joined me at that time. He covalently conjugated these with the carbon nanosphere, this molecule, which could activate the acetylation in the cell. This conjugated molecule could cross the blood-brain barrier. It could go to the brain. Not only that, by 3 days' time, it is full in the brain after subperitoneal injection. And we find that in the day 3, there is a huge acetylation in the brain. We could detect that. And this is a very specific acetylation we find in one of the very important acetylation, which often gets down-regulated, which is called histone H2B tetraacetylation. And that is becoming a very encouraging. So we ask the question, in the brain, which are the different parts this acetylation is upregulated? And what we find that the predominant acetylation we find in the dentate gyrus area of the brain, which is very important for neurogenesis and the cognate brain function. Of course, we also find in the cortex region. So we find that this is the CSP and this is the CSP TTK21, the molecule which activates the acetylation, is predominantly hyperacetylation we observe, like H4K12 acetylation or H2B acetylation, which is often down-regulated in the brain. So we ask the question, what it is doing by hyperacetylation? One possibility, if it induced the acetylation, is it affecting the neurogenesis process or dendritogenesis process? Very encouraging thing what we find that if you see the bottom here, the CSP in the dentate gyrus area, this is the dendritic growth. But here you see the CSP TTK21. Here it is inducing the neurogenesis, dendritogenesis, a huge manner. This made us to ask the question that if this is the case, is it affecting the memory function? We use the Morris water bath test, where the mice has a 4 marker here. There is a water bath, and this is the stage kept here. And first day mice take long time to reach to there. They don't like the cold water. Second day it remembers and go. Third day it goes straight there. And even if you go and give this stage, these mice, if they remember, they will be here only. By looking this propensity of the mice traveling here, you can make out the memory is there or not. To cut a long story short, the mice which got CSP-TDK21, they could remember this stage after 16 days even, and that shows that the CSP-TDK21, which is inducing the acetylation in the brain, is actually inducing the memory in the long-term memory. And so we look into the genes it is activating, we go on to the gene promoter, we show that yes, acetylation is directly connected to the overexpression of these BDNF and other genes. But this is with the normal mice. The beam what we started with that, can we do the reversal of the memory of diseased conditions? We took the Alzheimer's model system. I will not go through the Alzheimer's disease process here because of the time, but you all know it's a neurodegenerative disorder. It happens with the age, but nowadays it's starting much earlier because of the social, lot of stress, because of a lot of pollution probably. And it is only very small percentage is genetic. Most of them are sporadic, and it is behavior related and also food and exercise related. So what we took is a tau mice model, which is Alzheimer's model. And as you see here, this is the saline treated wild type mice. This is the tau mice where the neurodegeneration has started. And you see this is the CSP. And when you put the drug, you can see there is a repair in the neurons. Most importantly, we see the dendritic prime, which is actually upon learning, these small mushrooms forms in the dendrite. And these mushrooms are actually indication of the memory. And you see here, these are the mushrooms on the wild type. The tau mice, these mushrooms are very, very less upon learning. But if you give the drug, if you give the CSP-TDK21, the mushroom formation is almost the wild type, suggesting that this may be working to induce the memory even in the tau mice. We did a lot of analysis of the genetic analysis. We see that genes it is activating, very important 81 genes it is activating. It is actually, we generally do the ChIP-seq analysis with the acetylation mark, H2B tetraacetylation. We do find around 600 marks are upregulated. It is activating the super enhancer, 14 super enhancer. But the major question is it inducing the memory? Just to show you one data here, that this is the wild type mice memory. This is the mice got, this is the tau mice memory, and this is the mice which got the CSP-TDK21. So as far as memory is concerned, we could completely recover the memory. Of course, these mice were younger mice. Now we have started working with the very, very old mice where the disease has progressed a lot. And we have completed this for different reasons. I will not leak out the data, but there are very promising data even in the old mice. The point is, here, this is a new target that without affecting anyone, even if you give 5 years a better life, and you will be very, very encouraged to do so if it is been done in a non-toxic manner. Now, the CSP-TDK21, we can now don't need to give peritoneal injections. We can give the oral delivery, and it is still working. So we are in the process. I just end with the last 5-6 minutes, a serendipitous invitation and what it leads to based on this CSP-TDK21. When we are working all these, one of the, from Imperial College London, one of the scientists, Simon Gormley, he came to us and saying that your molecule is inducing the CBPP300 function. Here, I have a very interesting observation of CBBP3 and a spinal injury, repairing of spinal injury. I'll show you that last part. Spinal injury is a very terrible thing. It largely makes the paralysis for the rest of the body. It can happen to anybody at any time, and especially these are accident-related one. It is keep on increasing. So many celebrities are actually down today. So what was Simon's finding? This is Simon Robini from the Faculty of Medicine Department, Imperial College London. Simon finds that mice which has an enriched environment, okay, this is the enriched environment, they are happy, they're all this. Their CBP expression goes up. And in these mice, if there is a neural injury, they are repairing better. And so he told me that your molecule can induce the axonal growth. So can we use these for the spinal injury repair? Well, he tested our molecule and you see this is the CSP and this is what he put CSPT in neurite culture. And it branches like this in no time. So this gives an idea that if you have a neural injury like this, the spinal injury, then if you put CSP TTK21, he indeed sees that it is getting connected because of the axonal growth. When he did the functional analysis, he finds that indeed this is actually that the electric connection is happening. Then he did a couple of tests, okay? And I will just show you a video of what test we did. So this is called the ladder work. So if the spinal injury is there, these mice will be, the slippage will be more. If the injury is repaired, the slippage will be less. It could be having another test, which is called the gum test. Gum on the mice, on the foreleg, and it will try to open it up. And if the time it takes, if the spinal injury is there, it will take longer time. But if it is repaired, it will take less time. So what Simon finds that, what he finds that if you give this drug, the grid work, the injury gets repaired, and the grid work slippage comes down. And mice also take much, the contact of the tape is take much lesser. So it is functional. It is working. But seeing is believing. He created a very interesting video I could share with you. And I will show you 2 times in case you missed the first time. You see, this is the mice got the drug. Same injury time. It is working very nicely. I repeat this one. And the top one is got only the nanosphere, not the drug. You see, it is not been able to put the leg properly. So this shows that it is being able to repair the spinal injury very, very efficiently. Soon after this work got published in Science Translational Medicine, it became a cover also. I start receiving so many phone calls, so many mails. A voluntary organization from Singapore visited CDRI and they want to support this work to take to the next level. And this is the parents' organization or relatives' organization. Each one of their family member is somewhere or the other is got a spinal injury. Now we are trying to do the preclinical work in CDRI on this. We are hoping that this molecule will see light of the day, and this is how the serendipitous work and the fantastic collaboration can lead to a more meaningful science. So at the end, I acknowledge my colleagues, my collaborators. Some of their work I have not shown. I especially mention name of Dr. Ishan Murthy, Professor Ishan Murthy from JNCASR, and Annie for the different work, and of course Simon for the spinal cord work. Cancer works all are collaboration with Dr. Gopinath from SCG, Rishma from the NUS. Thank you very much for your kind attention.
Professor Shah, please.
Yes, thank you so much, Dr. for this wonderful exposition about your work. And I understand that there are a lot of collaborators are also there, and there are
Yes.
many intricate questions that may be asked. And I was wondering about the PCAP inhibitor that you alluded to in between. That what
Yes.
happened? You talked about it in the context of cancer, but we have so many muscular diseases like multiple
Yes.
sclerosis, muscular dystrophy, ALS, simple myopathy. So how, I mean, is it known that what kind of, what happens to PCAP in those kind of conditions?
It's a very, very important question. Actually, Dr. Risma Taneja's group is looking into it. And last discussion we have, there are another molecule, another enzyme like G9A, which is also involved in the, this muscle synthesis process. So now this exact data on the PCAP context of the muscular dystrophy and other muscle-related disease. Exactly, I do not know. Apparently, there is a effect, and she's addressing that.
Okay. So, yes, Dr. Nautil, did we get any
Yeah.
question? Yeah.
No, but there is a general question which I want to ask is, what you said was that genes will not be affected, but in diet, environment, and psychological factors, they can influence the epigenetics. So
Yes.
are these later changes reversible, or like genes, they are irreversible?
Yes, it is reversible. It is reversible. It is reversible, and but some of these are very difficult to reverse. Especially, people have shown that the methylation, especially the DNA methylation, is one of the. It takes long time to be reversed, and that needs a constant persuasions. And it also has a connection with the food, because you see most of the food, it has a different degree of SAM production, adenosine methionine production. So so the people are thinking that food and some sort of a, it comes back to what people throughout the time people have thought, the process of yoga, process of meditation, what value it has. So it is now evident that these process directly connect to the epigenetic modifications, and it has a value. So the reversal is possible through the food and the exercise and the meditation. But all cannot be changed. All cannot be changed. But yes, there is a reversal is possible. That was the purpose of showing that small video.
Yeah. Yeah. Yes. Can
There
yeah,
are in
there
the
are
chat box, I don't see.
I have we have a question from Dr. Madhav Bamji, and she asks that do microbiome act epigenetically also?
Absolutely, absolutely. Microbiome, actually the microbiome of different kinds of microbiome, it alters the metabolism, it alters the cofactor availability, and it has a very distinct effect in the epigenetic modifications. So there is a good microbiome, bad microbiome. There is a microbiome related to age and exposure to pollutions. All has the final effect comes from the the epigenetic alterations. And this is a huge field. People are just started exploring it.
Yes. Yeah. If we yes, Dr. Nautil.
Yeah, I don't see questions in the chat box. But has there been any practical application of CSP PTK21 so
Yes,
far?
we have not see it's a long route. We have just started the preclinical work, and so we are doing it. And the first thing probably we will be trying in the in the primate applications, or maybe in the permission from the from proper authority, the spinal injury one, TSPTTK21, and we are trying to see that within 1 year time we can reach to that level.
Thank you.
Because the spinal injury has a very direct effect, and the thing is, you don't have to continue for a long time. And in the animal model, in the three different animal model, we could repair it by 4 weeks time.
Okay. So I would like to thank all of you and ask Dr. Mandal to give the vote of thanks if we have no more questions.
Dr. Mondal, we can't hear you. I think you are mute.
Thank you. Thank you, Professor Kundu, for this mesmerizing, engrossing, informative talk. We are proud that you have agreed to deliver this first talk in the 75 years of independence AMRUTOTSAVA webinar series. All the viewers are definitely enriched by listening to this very informative talk on epigenetics. I sincerely thank President INSA for starting this series and her valuable guidance and kind support for conducting this webinar. I am thankful to Dr. Aurora, Dr. Nubia, and Mr. Jokarkar for their kind support. At last but not least, I thank Mr. Karthik, Mr. Aditi, Mr. Manish, and Mr. Ravi for their technical help for making this webinar successful. Thank you so much and stay safe.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you. Thank you. Stop.